Developing a treatment for cancer has been one of the biggest missions in the history of science and medicine. Research has been conducted on multiple methods believed to be the end-all treatment for this malicious disease, but none have yielded the results that were expected or hoped for. Gaining a better understanding of the physiological functions involved in these speculated treatments will save scientists time, energy, and resources by expediting the process of affirming or disproving the efficacy of possible cancer treatments. YoungerUSA holds the tool necessary for revealing these physiological functions that will be the key to finding the ultimate cancer treatment. Non-invasive micro-testing technology, or NMT, provides the ability to detect and measure the real-time flux of selective ionic and/or molecular activities en situ in live samples ranging from single cells of 5 micrometers to tissue and whole organs, as well as observe activities of tumor microenvironments. In recombinant horseradish peroxidase variants for targeted cancer treatment, published in the Journal of Cancer Medicine in 2016, the enzyme horseradish peroxidase, HRP, combined with indole-3 acetic acid, IAA, was researched as a powerful targeted anti-cancer agent. This was the first study describing the successful use of recombinantly produced HRP for targeted cancer treatment, and this discovery might lead to an increased use of the powerful isoenzyme HRP-C1A in future cancer research. This study focused on single tumor cells. In order to advance further, it is also necessary to observe the activity of IAA within the tumor microenvironment. By limiting the span of observation to single cells, other factors such as signaling molecules, immune cells, and blood vessels that surround the tumor are ignored. These aspects need to be considered when observing a tumor, as evidence shows they have substantial influence on the behavior and biology of the cancer. Observing the effects of IAA, HRP on the tissue level, the tumor as a whole, and the interaction between the tumor cells and their surrounding environment would provide significant progress in this research, as it would reveal deeper functions involved in this phenomenon. The types of treatments studied in this paper are gene and antibody-directed enzyme prodrug therapy, which allow the selective release of a cytotoxic agent from a non-toxic prodrug, in this case IAA, at the site of a tumor. With NMT, the activities and functions of IAA can be studied as the interaction between the treatment and tumor occurs in real time. NMT can provide crucial data, including which part of the tissue is responsible for the uptake of IAA and where the conversion occurred, as well as the concentration and location of endogenous IAA in and around the tumor. NMT could also be used to observe the effects of blocking ion transportation sites, which would provide proof of how IAA HRP affects cancer cells and tumors. Thanks for watching. See you next time.